ASRB net plant pathology preparation model question paper 18. I'm Manoj Shankar Reddy, PhD plant pathology. So the first one is Plasmodiopora was discovered by. Actually, the genus or maybe the life cycle and all those things was actually discovered by Tillett or H.M. Ward, Voronin or Anton D. Barry. So the Plasmodiopora brassicae, that is a clubroot of crucifers or cabbage, was first discovered by M.S. Voronin. He is a Russian scientist. He is also a student of Anton D. Barry. So around in 1870s, this clubroot of cabbage was a very big problem when it comes to Russia. So the Russian government openly said that whoever will identify the causal organism along with the management practices, they will get a good prize. Then in 1870s, where the Voronin started working, around 1874 he came to almost a significant proof, and 1877 he published the complete proof along with the management practices as well. So but this Plasmodiopora life cycle was discovered by Voronin. The next one is Coltotrichum falcatum causes anthracnose of chili, red rot of sugarcane, citrate of sugarcane, and anthracnose of pepper. So generally Coltotrichum falcatum is causes red rot of sugarcane. So anthracnose of chili is Coltotrichum capsicii and other species also there. So like this, so generally the answer is Coltotrichum falcatum causes red rot of sugarcane. And while studying this kind of MCQs, I also suggest you to, if one is the answer, the remaining three might be the answers for some other questions. Four options are there. One option is the answer. What about the three options? There might be the answers for some other questions, right? So if we correlate and start studying in such a way, that will be very easy. So in studying 100 questions, we will also get benefit of another 300, so that 100 plus 300, 400 questions at a time we will get, right? So that will be easy method also, and a little bit a time consuming process, but definitely it will yield you fantastic because I actually followed the same method. So the next one is a surface disinfectant, sodium hypochlorite, mercury chloride, water, and both A and B. Here sodium hypochlorite 1%, mercury chloride 0.1%, both can be used as a surface disinfectant. Here the concentration is very important. If it is a sodium hypochlorite, the concentration is 1%. If it is a mercury chloride, the concentration is 0.1%. Next one is a black mold fungus is generally refers to Penicillium digitatum, Rhizophus oryzae, Aspergillus niger, Aspergillus flavus. So generally the black mold fungus is generally refers to Aspergillus niger. Other groups of fungi like in Penicillium also in like generally Rhizophus and other species are generally also considered as some other species also generally considered as black molds. But when in general, if we give overall in general Aspergillus niger is considered as black mold fungus. And regarding this various kinds of molds in Aspergillus and various kinds of molds in Penicillium, in the previous videos we have published a very detailed structure about this various kinds of molds. Next one is the heat sterilization period at 120 degrees Celsius in the hot air oven will require how much amount of time? 1 hour, 2 hours, 3 hours, 4 hours. So the answer is 2 hours, because when the temperature increases, the sterilization time goes down. Here I have given for our understanding this is the various kinds of temperature that the time and the temperatures I have given it. If we are putting at 120, we need to sterilize at 8 hours, around 480 minutes. If at 140, 3 hours will be required, 180 minutes. If we are sterilizing at 160 degrees Celsius, 1.5 to 2 hours generally. At 180, we used to kept at 30 minutes to 1 hour. At 190 degrees Celsius, we used to kept at 16 to 12 minutes. So they asked at 120 degrees Celsius how much time it will require, around 1.5 to 2 hours. So there is no 1.5 hours in the option, so the answer is 2 hours. And remember, so sometimes we also need to adjust the answer along according to the options given. So don't think that whatever you study the same thing that will appear. So sometimes they may also give like more or less similar. For example, I will tell you one of my experience. For example, you can take autoclave temperature. It is exactly 121.6 degrees Celsius for 15 minutes at 15 psi pressure. But in one of my I remembered something that in one of my previous exam that I wrote somewhere else, I'm not recall it properly, but they have given 121 degrees Celsius for 15 minutes, 15 to 20 minutes at 150 psi. So the answer is this. So, but what we used to study is 121.6, 15 minutes and 15 psi only. but there is just no such kind of options will be there. So whatever it is close to that, we need to move according to that. So the options also very very important, so that you guys also just uh, touch up with the options that I have given in the exam. The next one is sixth one is the asexual spores produced by the many fungi. So generally, what is the asexual spores that is actually produced by the many kinds of fungi: conidiospores, ascospores, zygospore, basidiospore. Ascospore, basidiospore, and zygospore are the sexual spores, right? There is only one asexual spore, that is conidiospore. So majority of the ascomycota fungi that can produce conidia as a asexual spore, right? And basidiomycota group of fungi also can produce conidia, right? Next one, fungi causing downy mildew disease are differentiated on the basis of. So this downy mildew fungi are actually differentiated on what basis? First one is sporangiopore character, teleospore character, appendages attached on the cleistothecium based on the spore production. Here this downy mildew actually comes under oomycota. Teleospore, appendages, cleistothecia, the spore production is not at all related to this oomycetes group of fungi, where this oomycetes group of fungi contains sporangia and all those things, right? So here actually this downy mildew group of fungi are characterized based on the sporangiopore character. So how the sporangiospore branching, if I want to say the branches are the germination type, how it is germinating, dichotomously branched, or like what are the different kinds of branches will be there, the sporangiopore characters. Or here the downy mildew is actually characterized based on the sporangiopore character. It is a very important one. Gene for gene hypothesis proved on which crop? Rice, barley, linseed, or tobacco. So here the gene for gene hypothesis was given in a linseed. This linseed is also known as a flaxseed. This gene for gene hypothesis was first given by H.H. Floor in 1946 in a linseed rust or flaxseed rust, which is caused by Linum mysorens. So Linum isutatinum, Linum isutatinum. So this gene for gene hypothesis, which was actually discovered in flaxseed or linseed, discovered by uh, H.H. Floor in 1946. A typical long-celled rust fungi, next one, produces during its life cycle. How many spore forms can be produced? A typical rust. So we know very well, you can take an example of a wheat stem rust. How many spores it will produce? As 5 spores, right? Pycniospores, Achiospore, Iridospore, Teleospore, Basidiospore. So a typical rust contains 5 spore forms. A long cycle rust contains actually 5 spore forms. Of course, you can take some rust, they may be lack of 1 or 2 stages. That's why it's called as microcyclic. All the 5 stages are not present, right? So cedar apple rust or maybe some other kinds of things, 1 or 2 stages may be present. So if uridial stage is absent, it is known as the demicyclic rust. Demicyclic means lack of uridial stage is known as a demicyclic rust. So such kind of things also will be there. Generally, the a typical long cycle rust generally contains 5 spore stages. The next one, example of mycoherbicide: Coligo, Divine, Bimol, all the above. So what are all the options given? All are the mycoherbicides only. So the answer is all the above. Here this Coligo is the first mycoherbicide, but the first registered mycoherbicide is Divine. So all are mycoherbicide only, but the first mycoherbicide is Coligo. Registered means Divine. So this was actually first registered in USA, Divine. Next one is the leaf shredding and green ear symptoms are noticed in downy mildews, wilts, rust, and smuts. We know very well, in especially cereals, we can see this sorghum downy mildew, maize downy mildew, such kind of downy mildew we might see, right? So generally, this is actually the downy mildews. Other names are like if it is a maize downy mildew, so crazy top, such kind of other names will be there. This leaf shredding or green ear symptoms are generally the characteristic symptoms of this downy mildew. So the answer is here, downy mildew. The next one is bean anthracnosis caused by. It's a very 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 important pathogen. This is a bean, imagine. So we can see circular black color dot. Simple example I'm telling. I'm not explaining all the symptoms here. Like depressed black color spots or sunken spots can be seen on the infected fruits and sometimes in the leaves as well. But the fruit symptoms are the most diagnostic and characteristic symptom and important also. So it is caused by Collotrichum lentimontianum, Collotrichum glaceproides, Collotrichum capsicum, Collotrichum graminicula. So beans anthracnosis is caused by Collotrichum lentimontianum. It's a very very important one. So the next one is pierced disease of grapevine is caused by Irvinia amylovera, Xylella fastidiosa, Pseudomonas solanaceae, Xanthomonas vitis. So pierced disease of grapevine is generally caused by Xylella fastidiosa, and this other disease also you guys just go through. Next one is measuring the size of minute objects in a microscope is known as pathometry, micrometry, turbidometry, hemometry. So generally micrometry is a separate branch. Generally nowadays we have a microscope that is equipped with this micrometry techniques, like just measuring the minute objects. So such kind of minute objects can be measured with the help of micrometry. So the equipment, like it's like a scale only, it's called a micrometer. Okay, so generally we use this technique, or this method is the micrometry. Fig smut is caused by Aspergillus flavus, Aspergillus figi, Aspergillus niger, Puccinia striformis. Here fig smut is caused by, generally smut means we may thought that it is caused by a smut genera, maybe Istulago or some other kinds of genera, but it is generally caused by Aspergillus niger. Aflatoxin dangerous form. What they're asking is which form of the aflatoxin is more dangerous, B1, B2, M1, M2. So to have an understanding about this, there are few strains that Aspergillus can produce. If you see, Aspergillus flavus produces B1 and B2. Aspergillus parasiticus produces G1 and G2. Aspergillus nominis produces M1 and M2. So among all the strains that is produced by the Aspergillus species, B1 is the most toxic and most dangerous form. So the answer is A, B1. Phloem necrosis of coffee is caused by Phytomonas leptovesorum, Phytomonas teheli, Phytomonas francis, Phytomonas bancrofti. So here this phloem necrosis of coffee is caused by generally Phytomonas leptovesorum. I was a little bit doubted about this. You just guys just please go through. And the next one is the lead blight of potato is the best example for endemic, epidemic, epiphytotic, pandemic. Here this lead blight of potato is the best example for epidemic disease. Epidemic is otherwise called as epiphytotic also, and is also an example for pandemic disease. So the answer is actually both B, C and D. But if such kind of cases occurs, I would like to suggest to go with pandemic, because this is the best example for pandemic which is present everywhere in the world, right? Of course, it is epidemic in Ireland in 1845 and 1917 in the Germany. So, but it is the best example for pandemic diseases. If such kind of options prevail, I suggest you to go with the pandemic. It's your own decision, but I will go with the pandemic. The genus of powdery mildew pathogen that produces both external and internal mycelium is known as. So to understand again the powdery mildew, on the right side I have given some pictures. These are all the types of powdery mildews: idiom type, oidiopsis type, ovular type. Idiom type is example. So for example, before explaining this and all those things, I will tell you, if you see in idiom type, the mycelium is outside. In oidiopsis type, the mycelium is inside, which goes internal mycelium. When it comes to ovular type, the mycelium is both external and internal. So idiom type example is Ericife. Oidiopsis type example is the Levilula. Ovular type example is the Phylactinia. Now the answer is both internal and external, only one kind of mycelium is present. Ovular type example is Phylactinia. So the answer is D, Phylactinia. So regarding this types of powdery mildew, on the next slide I am going to explain what all the different types of powdery mildews are there. The next one, blast disease of rice was first reported or recorded in Japan, China, Philippines, India. Actually, the blast disease of rice was first reported in China in 1637. It is a very old recorded disease in China. Of course, other diseases also there, but it was first reported in China in 1637. In India, it is reported in around 1918, I think, in Tamil Nadu, Tanjore district around, because the Tanjore is very famous for the production of rice, right? So in there, this disease was first reported in India around 1918 and 1919 time. The Padmanabhan, the scientist who identified this blast disease for the first time in India, and this blast become very severe in around 1985 and 86 in same Tamil Nadu region. So, but the answer for the question is China, 1637. So these are all the different types of powdery mildews: idiom type, oidiopsis type, ovular type. So when we see here, the symptoms are mostly appears on the upper surface if it is idiom. So if it is oidiopsis, lower surface. If it is ovular abscess, both surfaces the symptoms will appear. So the mycelium, the next one is, so the mycelium is generally hyaline, septate, ectopytic. Before question we have in the previous slide we have seen ectopytic. So it is a endophytic. Ecto means outside. The mycelium is present. Ecto means outside in oidium. Endophytic means inside. When it comes to ovular abscess, the mycelium is both ectopytic, that is hyaline, both ectopytic and endopytic. Haustoria, yes, epidermal haustoria only present. Haustoria is actually sponge like annulus present, but epidermal haustoria is present in ovular abscess type. Conidiopore is actually short club-shaped non-septate. When it comes to oidiopsis type, long branched septate. Ovular abscess is branched but septate. And conidia. Conidia is generally cylindrical or barrel shaped, which are in chains. We can see here cylindrical, barrel shaped, which are in chains. But when it comes to, it is actually a club shaped. You can see here club shaped. And when it comes to conidia in ovular abscess type, single celled clavate or maybe club shaped, clavate shaped or maybe sometimes club shaped also. Generally, Cleistothecium contains mycelioid appendages. All powdery mildew generally produces Cleistothecia, right? This Cleistothecia contains mycelioid appendages. In the previous lectures, I have explained what is mycelioid appendage, what is bulbous appendage, all kinds of appendages I have explained in the previous slides. For your reference, you may just look into the previous videos. And again, the oidiopsis also produces mycelioid appendages. Ovular abscess produces bulbous appendages. And coming to host, the best example for oidium is a Hericife, oidiopsis is a Levilula, ovular abscess is a Phylactinia. And these are all a couple of examples. Grapes powdery mildew, black gram powdery mildew, apple powdery mildew, rose powdery mildew. These are all the best example for oidium type. Chili powdery mildew and PGNP powdery mildew is the best example for oidiopsis type. Whereas the mulberry powdery mildew is example for ovular abscess type. This is about the powdery mildew genera. So the next one is blotter test is used in seed testing, seed viability, seed purity test, and the last one is a seed germination test. Generally, this blotter paper is used for a seed health testing, and it was actually developed by Louis Dyer. I think I'm not sure, probably Louis Dyer. And the next one is a blotter test is commonly followed for the detection of seed-borne viruses, seed-borne bacteria, seed-borne fungi, and seed-borne nematodes. Generally, this blotter test is recommended for the identification of seed-borne fungi, or maybe any kind of mycelium, or maybe some kind of resting structures or spores if they are present with the moisture that may germinate, so that we can easily identify. Seed-borne viruses and bacteria we cannot detect by using this blotter paper method. Only seed-borne fungi we can detect by using this blotter paper method. The next one is a host plant on which wheat rust fungus produces teleal stage is known as, I repeat once again, host plant on which wheat stem rust fungus produces teleal stage. There are 5 stages are there: pycnium, acium, iridium, teleum and basidium. What they are asking, teleal stage is produced on which crop? Alternate host, primary host, collateral host and barberry. Here I have given pycniospore, aciospore, iridospore, teleospore, basidiospore. So generally the stages, germination, color, everything I have given, since I don't want to waste time. Here the teleospores produces on primary host. Primary host means wheat. So both the teleospores and iridospores produces on the primary host upper surface. Here pycniospores and aciospores produced on the alternate host, that is barberry. Pycniospores produces on the upper surface, aciospores produces on the lower surface. But when it comes to iridospores and teleospores, both produced on the primary host, the two upper surface. Okay. The next one is significant contributions on the epidemiology of disease cycle of stem rust of wheat was made by K.C. Mehta, Munkur, Paik, Daster. The first person who actually started studying this wheat stem rust in India is K.C. Mehta. That's why he is known as father of Indian rust. So the answer is K.C. Mehta. So the next one is the sexual spores produced by fungi belonging to Deuteromycotina, ascospore, oospore, no sexual spore, zygospore. Actually, this Deuteromycotina is also known as fungi imperfecti or dustbin fungi or some other names. Generally, these two names are very famous: fungi imperfecti and dustbin fungi, which means it contains no sexual stage and it produces no sexual spores. So the answer is no sexual spores. Next one is 26. Fungi with a dichotomously branched sporangiospore at acute angle with curved pointed tips on the sporangial bone. There are 5 types of this downy mildews are there: plasmopora, bremia, and peronospora, such kind of things that I have explained in mycology videos, previously published mycology videos, along with the previously published net videos. We spoke in detail about this, especially this net related videos section. If you go to mycology, in the mycology section net related videos, we spoke detail about this various kinds of downy mildew. Now I'm just telling the answer. So the answer is peronospora. Okay, peronosporum produces simply acute angle with the pointed tips. Next one is Elisino ampelina causes anthracnose in which crop? Elisino ampelina causes anthracnose in citrus, mango, strawberry, grapes. Grapes. Generally, this Elisino ampelina causes grapes anthracnose. This grapes anthracnose otherwise is known as a bud eye spot of grapes, which is caused by Elisino ampelina. Then repeating spore of rust. So teleospore, basidospore, aesiospore, and iridospore. Generally, iridospore is the repeating spore, teleospore is the resting spore of rust fungi. Iridospore completely repeats, that's why it's called as a repeating spore. Teleospore is a resting spore. Okay. Next one is the answer is iridospore. Frog eye spot of tobacco is a serious disease which is caused by. They're asking frog eye spot of tobacco is caused by Cercospora moricola, Cercospora nigricans, Cercospora lycopersicum, Cercospora nicotianum. So the answer is Cercospora nicotianum. Next one, brain flotation method is followed to control loose smut of wheat, Tundu disease of wheat, grain smut of jowar, ergot of wheat. So generally this brain flotation method can be recommended for the management or for the control of wheat Tundu disease. The perfect stage of Helminthosporium varese. Generally this Helminthosporium varese is caused by rice brown leaf spot disease, otherwise called as Bengal famine, right? Helminthosporium varese, otherwise called as bipolaris varese, otherwise called as Drusillera varese. So the answer is B, Drusillera varese. Cochleobolus miabilis also we can say. But here the options given is the Drusillera varese. So the answer is Drusillera varese apart from the other options, Sakari and Haloides. And the next one is a toxic substance that is produced by the Trichoderma viridi. So glyovirin, viridin, subtilicin, pseudobacterium. So this subtilicin, bacillus subtilis, pseudobacterium, maybe pseudomonas. Glyovirin, I'm not sure, probably some species of pseudomonas, I'm not sure. The next one viridin. So the answer is viridin. So Trichoderma viridi produce various kinds of toxic compounds or substances, viridin, viridicatin, and such kind of substances are produced. But the most important thing is viridin. Next one, Cleistothecium containing several asci bearing a definite rigid appendage with coiled tips. Very simple. If coiled tips are there, it is uncinilla. So regarding this Cleistothecial appendages, we have already published before. Now if coiled tips comes, it is uncinilla. Or curled tips, sometimes they may also ask in such a way for confusing us. So the coiled tips or curled tips means uncinilla. International Mycological Institute is located at England, USA, Netherlands, India. So this International Mycological Institute is located at Kew, England, which was started in 1920. The first International Mycological Institute, the director of first International Mycological Institute is E.J. Butler. So the answer is England, Kew, started in 1920. Fungi causing smut diseases belonging to the order Ascomycota, Basidiomycota, Deuteromycotina, Zygomycotina. So generally this fungi causing smut disease belonging to the phylum Basidiomycotina. All rusts and the smuts comes under this Basidiomycotina. Next one, diseases which interfere with the conduction of water in plants: wilts, rots, leaf spots, damping up. We know very well vascular wilt disease or wilt disease generally obstructs with the passage of water in xylem and phloem vessels, so that there is no translocation and connection to the roots and shoots, and there is no translocation of water from roots to shoots. That's why it starts wilting, right? So the answer is a wilting. Toxin produced by Fusarium causing vascular wilts: Fusaric acid, fusicokin, pisatin, zearalenone. This pisatin is actually a phytotoxin that is produced by P. Zearalenone is a toxin, it is also produced by Fusarium. But the most important thing when it comes to vascular wilt diseases, the Fusarium producing toxin is known as fusaric acid. Very important one. Next one, genus Alternaria belonging to family Tuberculosis, Moniliaceae, Dermataceae, Pleiosporaceae. Probably Pleiosporaceae. I'm not sure. I believe maybe Pleiosporaceae. I'm a little bit doubt about Moniliaceae also. Probably Pleiosporaceae. Please check once. And first bacterial A virulence gene was discovered in which crop? So tobacco, tomato, soybean, mung bean. The first bacterial A virulence gene was discovered in soybean. I think soybean. Bacterial wilt have soybean, Pseudomonas syringae pathovar lacrimens, Pseudomonas syringae pathovar syringae. I think so, I'm not sure, but the crop is soybean only. Around 1984, I think it was the first discovered in. The answer is soybean. The first discovered bacterial evolution is soybean only. In fungal classification, class ends with. So in fungal classification, there is some hierarchy is there, right? Always ends with what? For example, if you phylum always ends with Mycotina, Mycetes. Class always ends with Mycetes. Class always always ends with. Generally, in hierarchical classification, phylum ends with Mycota. Class always ends with Mycetes. Order always ends with Yales. So such kind of things will be there. Regarding further more clarification and further more information, you guys can refer our previously published videos, especially classification of fungi and taxonomy of fungi, where we have mentioned in detailed classification and what where the classification started, what are all the different kinds of classification are there, how many classifications are done, what are the present classification that we are following. In mycology section, if you go to playlist, there is a separate section called mycology, and there is a video called probably taxonomy and classification of fungi. There we have published all those things very in detail about. Next one, which of the nutrient is supplied by mycorrhiza to the plants? So generally this mycorrhizal fungi supplies phosphorus. So it is a very important nutrient to maintain the disease resistance against various kinds of pathogens. So the answer is phosphorus. And the next one is a fungi belonging to the order Eustylaginales are known to cause rust, ergots, smuts, all of this. Generally, Uridinales causes rust, Eustylaginales causes smut diseases in crop plants. So this rust and smuts are comes under Basidiomycota group of fungi. Next one, whip smut of sugarcane is caused by Stylago noda, Stylago hardy, Stylago urocystis sepulae, and Stylago citaminium. So Stylago citaminium generally causes a whip smut of sugarcane. Now the name changed to Sporisorium citaminium. So before it is called as Stylago citaminia, now it is called as Sporisorium citaminium. The next one is sporangium on germination produces vesicle on the genera. So for that I have given this picture. This is the life cycle of Pythium aphanidermatum. So on the sporangiospore, they form a small club shaped or form the small a vesicle like structure is club shaped structure is formed above the sporangium. So that the structure is known as a vesicle. So in the vesicle only zoospore formation or compartmentalization of protoplasm will takes place. If this protoplasm is present in sporangia, it remains as a sporangia. So this protoplasm starts to move into the vesicle where the compartmentalization takes place and zoospore formation takes place in the vesicle. Here the sporangium and germination produces vesicle in the genera, the special feature of Pythium aphanidermatum. The next one, osteolate ascocarp is known as, so which means osteolate, osteole presenting, in which ascocarp the osteole is present, they are asking. Okay, Cleistothecium is a completely closed ascocarp. Apothecium is a completely opened ascocarp. When it comes to perithecium, this one we can see is there is a small opening or slit or opening is known as osteole. So this osteolate ascocarp or osteole present ascocarp is known as perithecium. Next one, black spot of rose is caused by Cercospora species, Alternaria species, Diplocarpon species, Coltotrichum species. Generally, black spot of rose is caused by Diplocarpon rosae. Some other species also are responsible. That's why I have given Diplocarpon species. The subdivision ends with Mycetes, Ales, Mycetidae, and Mycotina. So in the previous slide also I have told that hierarchical classification, phylum, division, subdivision, all those things and detailed things that we have given in taxonomy of fungi. So the answer for the question, the subdivision is always ends with Mycotina. Next one, hermaphroditic fungi are those in which each thallus bears both male and female organs. Some thallus bears only males and some thallus bears only females. Sexually functional structures are produced in the morphologically distinguishable as a male or a female. So here the hermaphrodites means which contains both male and female organs. So same thallus contains both male and female organs. We can also call it self-fertile. So that such kind of fungi is known as hermaphroditic fungi. Next one is fungi is a member of the order Iridinales, Eustulaginales, sorry, not fungi. Puccinia is the member of the order Iridinales, Eustulaginales, Erysipelas. Generally, this Erysipa powdery mildews, Iridinales rust fungi. So Puccinia is rust. Eustulaginales generally all kinds of smut fungi. The next one is the teleospores of coffee rust pathogen. Here the answer is Iridinales. The teleospores of coffee rust pathogen is, first of all, we need to know what is the coffee rust is caused by. So the fungi responsible for coffee rust is the Hemilia vastastrix. Hemilia produces teleospore is a turnip-shaped teleospore and kidney-shaped iridospore. So in the types of iridospore in the mycology section we have mentioned the different types of teleospore. What is a single cell teleospore, double cell teleospore, multicellular teleospore, parachute-like teleospore, phragmidium, various kinds of things that we have mentioned in the mycology section. That is in the net related videos in the mycology section we have mentioned. If you want, you can also go and take a look into principles of plant pathology that we have published for UG level, but I suggest you to go with net related videos. And we are also doing mycology related videos now, where we are planning to the top to bottom in mycology section. So definitely this mycology will helpful for not only for semester related things but also for various other kinds of exams. So here the answer is turnip shaped. Next one, which copper fungicide is recommended for soil drenching? Bodo mixture, chestnut compound, burgundy mixture, all the above. So generally, burgundy mixture is recommended for soil drenching. I have no idea whether this chestnut compound or other things will be recommended, but this burgundy mixture is generally recommended for soil drenching. So the next one is monogenic resistance is also referred as. So in there are two kinds of resistance is there, vertical resistance and horizontal resistance. Vertical resistance is known as monogenic resistance. Why it is called monogenic? Because the vertical resistance is governed by a single gene. That's why it's called as monogenic. Mono means a single. Genic means genes. So monogenic, single gene. So horizontal resistance is governed by multiple genes. That is it's called as oligogenic. Oligo means multiple. So monogenic resistance is also referred as vertical resistance. Other names also have been given in the other related videos regarding epidemiology section. We have mentioned this. And the next one is heterogeneous nature of rust means a fungi requires one host species to complete their life cycle. A fungi requires two different hosts to complete the life cycle. Both A and B, none of the above. Hetero means different. Aeschus means general plants or maybe the host requires. So hetero means the pathogen requires more than one host or the two different hosts to complete the life cycle is known as heterogeneous. The best example for heterogeneous macrocyclic rust is the wheat stem rust. So heterogeneous means a fungi requires two different hosts to complete the life cycle. Best example is wheat stem rust. Next one is the powdery mildew of chili is caused by Erysipe polygoni, Erysipe cacaracearum, Levulla taurica, Sparotheca fuligina. Here chili powdery mildew is caused by Levulla taurica. The next one is the Phytophthora belonging to the family. Peronospororaceae, Pithiaceae, Peronospororaceae, Albuginaceae. Pithiaceae, Pithium, Albuginaceae, Albugo candidate, white list of Amaranthus. Peronospororaceae, generally this Phytophthora comes under Peronospororaceae. So the answer is Peronospororaceae. Next one, the spatial stage of Claviceps purpurea is known as honeydew stages, in which is full of conidia, ascospore, asci, and perithecia. So this Claviceps purpurea also produces sclerotia, but the spatial stage when it comes to produce abundant amount of conidia. So the answer is conidia. P. rust is caused by Puccinia, Euromyces, Pacospora, and Urocystis. Generally, this P. rust is caused by Euromyces appendiculatus. I think P. rust is caused by Euromyces appendiculatus. I'm not sure, but the genus is Euromyces. There is no doubt about it. And ascomycetes and basidiomycetes are known as higher fungi, lower fungi, imperfect fungi, dustbin fungi. So this ascomycetes and basidiomycetes are well-developed fungi, or perfect fungi, or higher fungi. We can take lower fungi. For example, oomycetes and zygomycetes are generally considered as lower fungi. Imperfect fungi means the fungi which lack completely sexual stages. We can call the Deuteromycota group of fungi, Deuteromycetes group of fungi is known as imperfect fungi. We can also known as dustbin fungi. In 2008 classification, this Deuteromycetes group of fungi also that was added to Ascomycota group of fungi only. Now it is actually under Ascomycota group of fungi. If you see the previous classification, Alexopoulos Mims classification, Ainsworth classification, they have a separate phylum called Deuteromycota, where all dustbin fungi and perfect fungi are placed under. Now in 2008 Kirk et al. Classification, this imperfect fungi was grouped under Ascomycota only. Mycology is the study of worm, algae, mushrooms, fungi. So again this is a little bit tricky because vesicular arbuscular mycor is also a fungi. Mushroom is also a fungi. Fungi is also so mycology deals with the worm, mushroom, fungi, all those things. Generally mycology is the study of fungi. So it's such kind of options given. I will go with fungi. Of course, general the definition of mycology is the study of mushrooms, or generally we used to call the study of fungi. So because mushrooms also fungi, like the before fungi, mushrooms are the first organisms that was evolved on the planet, right? So before the mushrooms are like very giant. Nowadays we have very little mushrooms. Before it was very giant, like trees, very big trees. So the mycology is generally study of fungi, mushrooms as well. So but I will go with the answer of D, fungi. So powdery mildew of apple is caused by Erisiphe polygoni, Podospora leucotrica, Sparotheca phylogenia, and Uncinilla necator. Here they asked apple. So apple powdery mildew is caused by Podospora leucotrica, Uncinilla necator, grapes powdery mildew. I think Erisiphe polygoni, it causes powdery mildew in black gram. I mean millets. Sorry. Uh, uh, no, yes, black gram powdery mildew, bendy powdery mildew, some kind of powdery mildew is actually caused by this Erisiphe polygoni. Sparotheca phylogenia, probably, I'm not sure, maybe cucumber powdery mildew, something like that. So actually this cucumber powdery mildew is caused by two types of fungi, I think, Sparotheca phylogenia, Podospora xanthi, and Erisiphe cicaracearum, Galvanomyces cicaracearum. Some things will be there, but I am not that much clear about it. Please go through, or if it is possible, I will try to mention in the next video. This generally here the powdery mildew of apple is caused by Podospora leucotrica. The next one is the germ theory of disease was started or originated with the work of Antonio Leeuwenhoek, Louis Pasteur, Theophrastus, Robert Koch. Actually, it was originated by Louis Pasteur and it was supported by Robert Koch, Anton Deberry, and various other scientists. But actually, it was originally initiated or started with the work of Louis Pasteur only. Germ theory of diseases. Raising the soil pH just above, or by adding the lime, that is calcium carbonate, gives very good control of potato what? Common scab of potato, powder scab of potato, club root of crucifers. Here the club root of crucifers is very severe in the acidic soil pH. If the soil pH is around 5.2 or maybe 5.5, this disease is very severe. So the best management practices raise the soil pH. Again in the first question itself, Plasmodiopora was discovered by Voronin. We have discussed it, right? So this Plasmodiopora, club root of cabbage, which is caused by Plasmodiopora brassicae. So it was very severe at acidic soil. The best management practice is raising the soil pH. How can we raise the soil pH? By adding the lime, that is a calcium carbonate. So around 2.5 tons per hectare of calcium carbonate if we add, we can raise the soil pH. That is the best management practice of club root of cabbage, because this club root is very severe in acidic soils. If we increase the soil to above just 7 or maybe 7.2, something like that, we can easily control the disease. So this raising the soil pH we use to control this club root. Next one, blight of taro is caused by Phytophthora palmivora, alternaria solani, Phytophthora colocaceae, alternaria alternata. So blight of taro is generally caused by Phytophthora colocaceae. Next one, seedling blight of castor is caused by Phytophthora parasitica, was first recorded and studied by. So there is a scientist JF Daster is well known and renowned for the establishing of a Phytophthora genera in castor, that is seedling blade of castor Phytophthora parasitica in India. So in India, this Phytophthora genera was established by JF Daster, and he is also the second president of IPS, Indian Phytopathological Society. This IPS was started by BB Munkur in 1947 February 28. Actually, later in come into enforcement at 1948 only, but it was actually started in 1947 February 28. So that was started with the chairmanship of SR Bose and BB Munkur. The first chairman or first president is SR Bose. He was worked very less time. Later JF Daster took the chair, and some people used to believe that JF Daster is the first, but if you see the IPS society or the IPS website, SR Bose is the first president. The second president is JF Daster, and is well renowned for this establishment of Phytophthora genera in India. So this seedling blight of castor is caused by Phytophthora parasitica. That is also very important one. And the dikaryotic mycelium is a distinct feature of the fungi belonging to Ascomycota, Diplomysticomycotina, Deuteromycota, and Basidiomycota. This dikaryotic mycelium is a very famous one when it comes to Basidiomycota. That is the most distinct feature or the most common feature of Basidiomycota generally. Then next one is green ear disease of Bajra or Kambu is also known as leaf shredding, crazy top, downy mildew. Generally, this green ear Bajra is known as downy mildew. Crazy top means maize downy mildew. Sorghum downy mildew is called leaf shredding. So this is the general way. I repeat once again. Leaf shredding means sorghum downy mildew. Crazy top, maize downy mildew. General downy mildew means green ear of Bajra or Kambu is known as general downy mildew. Okay. I think this is the same next question. Downy mildew of sorghum is otherwise known as leaf shredding, crazy top, both A and B, none of the above. A, leaf shredding. Downy mildew of sorghum is known as leaf shredding. Downy mildew causing pathogens, powdery mildew causing pathogens, and rust causing pathogens are obligate parasite, facultative saprophytes, obligate saprophytes, facultative parasites. They are highly obligate parasite because they cannot live without a living host. They are strictly obligate. They strictly requires a living host for their survival. So the next one is downy mildew of maize is also known as leaf shredding, crazy top, A and B. None of the just now we discussed. Crazy top is generally referred to downy mildew of maize. Next one, branching of sporangiospores in Plasma paraviticola. This Plasma paraviticola causes grapes downy mildew. To the main axis is acute angle, acute angle, obtuse angle, acute angle, right angle, unbranched. Generally, these types of downy mildew, if you see this plasmopora is a right angle. So right angle type of sporangiospore we can observe. Right angle means imagine this is the sporangiospore. So such kind of right angle with the blunt tips kind of sporangiospore we can observe in plasmopora. The term necrosis indicates death of cells, curling, blightening, atrophy. So death of cells is generally known as necrosis. So the answer is A. Next one, ring spot of sugarcane is caused by Stagnospora sacchari, Dimerella sacchari, Bipolar sacchari, Leptospirea sacchari. This ring spot is probably Leptospirea sacchari. I was a little bit doubted about it. Please refer yourself. So I believe it is Leptospirea sacchari. So kernel bunt of wheat was reported for the first time in 1931 by, this is the most repeated question. It was first reported by T. Mitra in 1931 in Karnal district, an agriculture station of Karnal district. That's why it is known as kernel bunt, which is caused by Tilletia indica. Later name changed to Neovetia indica by Munkur. Again the name is changed to Tilletia indica. Now scientists are all the literature. If you see Tilletia indica and Neovetia indica, both names are we are using. So it was first discovered by T. Mitra in 1931. So the kernel bunt infected fields produce a odor called some kind of fishy odor, some kind of foul smell it will produce due to production of a chemical called trimethylamine. So due to the production of trimethylamine from the infected grains, so the infected field produces a fishy odor or some kind of foul smell. Next one is a fusarium wilt of banana is also known as bunchy top, A and B, Panama wilt, and none of the above. This fusarium wilt is also known as a Panama wilt because it was first discovered in Panama. So the next one is the Molia disease of barley in India was first reported from Rajasthan by Prasad et al. In the year. So this year, I'm not sure, maybe 57 or 69, I'm not sure. Please refer this question. I believe it's maybe 57 or 58, I think, I'm not sure. Probably 57 or 58. In which country flag smut of wheat was first observed for the first time? Australia. So in 19 not in the starting of the 19th century, so 20th century, and exactly 1890s time, this flag smut of wheat is a very dangerous disease. This was introduced in India from Australia in 1906. So, but this was first reported in Australia around 1890s around at the time. But it was still it is a dangerous disease. But the severity of this flag smut now it was greatly reduced with the introduction of resistant varieties. So the ability of a pathogen uh, being able to cause the disease is termed as pathogenesis, pathogenicity, infection, and penetration. So the ability of a pathogen to cause the disease is known as pathogenesis. Flag smut, false smut of rice is caused by Neovacia horrida, Entaloma varese, Histologocetaminium, Histologonoida varians. So Entaloma varese generally called as false smut, Histologonoida varians of false smut. Histologocetaminium smut of sugarcane, Neovacia horrida, probably with some kind of disease, I'm not sure. The next one is the first Indian scientist who collected and identified fungi from India. So Barclay, one scientist was there. He's not an Indian, some other. But when it comes to India, the first scientist who collected and identified fungi is K.R. Kritikar in 1885. This was the most repeatedly asked question. It's a very important one also. The first Indian scientist who started collecting and identified fungi from India is K.R. Kritikar in 1885. The sexual spore of Rhizophus. So Rhizophus generally produces sporangia and as well as zygospore. This zygospore is generally a sexual spore. Zoospore, chlamydospore, aplanospore, and zygospore. This zygospore is the sexual spore. Imagine this zygospore. It is also contains some holdings like things that is called suspensor, right? Suspensor can be observed in iridospore. So this zygospore. So this is the zygospore, the sexual spore that is produced by the rhizophus, and it also produces planospores. Sorry, aplanospores. Then next one is vegetative spore of fusarium. So rhizoids, isomorph, sclerotium, chlamydospore. So generally fusarium produces three kinds of spores: microconidia, macroconidia, and chlamydospore. Here they asked a vegetative spore, so the answer is chlamydospore. Who used, actually it was a little bit faded out, who used a lens for the first times to study microorganisms? Robert Koch, Antonio Leeuwenhoek, Louis Pasteur, and T.J. Burrell. So the first person who actually after Zacharias Janssen, who extensively started working on microscope, is Antonio Leeuwenhoek. He not only discovered this compound microscope, developed compound microscope, but is also the first person who worked on magnification lenses. He developed more than 300 types of magnification lenses. So the used lenses for the first time in microbiology study was first used by Antonio Leeuwenhoek only. And still many reference books also mentioned that who discovered the first microscope is also Antonio Leeuwenhoek, right? So that microscopic lenses also, the magnification lenses also developed by Antonio Leeuwenhoek only. So the multi-celled spore in case of rust. So pycniospore, Asiospore, Iridospore, Basidospore, all are single-celled. In condition are dikaryotic in condition maybe. But if you see this multi-celled means more than one cell. Teleospore is a bicell, right? It is also dikaryotic in condition. But this is the multi-celled spore when compared to the other spore. All are single-celled spores, right? So the answer is teleospore. So the flag smut of wheat is caused by Histolygotritis, Histologo Histolygo Histolygonuda. Here flag smut of wheat is generally caused by Urocystis agropyri, ASRB. Next one is a PDA medium do not contains. PDA means potato, dextrose, agar. That's it. Dextrose is present, agar is present, potato, that is must and should. So beef extract is not present or do not contains or do not present in the PDA medium. So the answer is beef extract. Next one, select the wrong pair. Blackum of cotton, Xanthomonas compestus, Pathor malvacearum, bacterial leaflet of rice, Xanthomonas varese, Pathor varese, which causes bacterial leaflet of rice. Rice tripe of sugarcane, Pseudomonas rubrillians, and ergot of bajra. Generally, this ergot is caused by claviceps species, right? So here they have given Irvini, it is a bacteria. Ergot is actually a fungal disease. So the wrong pair is ergot of bajra. The next one, the perfect stage not seen: zygomycota, ascomycota, basidiomycota, deuteromycota. Deuteromycota, because deuteromycota is A fungi which produce no sexual spore, dustbin fungi or fungi imperfect we used to call, right? So the next one is a pathogen responsible for charcoal rot of soybean. All charcoal rot is caused by Macrophomina fasciolina only. With the closing eyes you can put it. So Macrophomina fasciolina. So the next one is apple scab is caused by Podospora leucotrica, Spilosia pomi, Venturia inequalis, Armillaria milia. This Venturia inequalis is generally called as apple scab. The leaf fitness period of apple scab is 9 hours. So to remember this, apple is 5 words, scab is 4 letters. Sorry, apple is 5 letters, scab is 4 letters. 5 plus 4 equal to 9. You can remember, we can remember in that way. But this leaf fitness period may varies with the environmental conditions and temperature. But generally the leaf fitness period is 9 hours, where these perithecia survive in the infected leaves. Venturia inequalis. Next one is application of potassium increases resistance to waterlogging, frost resistance, disease resistance, and none of the above. Generally, this application of potash have two benefits when it comes to plant pathology: disease resistance and drought resistance. So the option given is the disease resistance. So the answer is disease resistance. The iridospores of Puccinia graminis are disseminated by wind, animals, insect, and water. These iridospores are generally windborne, disseminated through wind. Teleospores of rust has gem pores in a number. How many number? 1, 2, 3, 4. Again, this number of gem pores that is produced by the Pycniospore, Asiospore, Rhodospore, Teleospore we have already given. But here the answer is 2. The teleospore generally produces 2 gem pores. What are all the three major factors in a disease triangle? Imagine this is a disease triangle: host, pathogen, and environment. Generally, these three are the very important factors. So, host, pathogen, and environment. Three factors are present, that's why it's a disease triangle. If four factors are present, adding time, it's a tetrahedron. Adding time, tetrahedron. If a human is added 5 factors, it's called a pyramid. So, which one of potato tuber rot is the potato tuber rot nematode? Didlingus angustus, Didlingus destructor, Anganatritici, Heterodera avini. Didlingus destructor is generally the potato tuber nematode. It causes general damages to potatoes. Number of tile fibers are present in T4 bacteriophage 1648. So generally this is the typical structure of T4 bacteriophage. So their parts, head, tail, all those things. If you see here a number of tile fibers 1, 2, 3, 4, 5, 6. Not only this T4, every T4 contains the same number of tile fibers, that is 6 only. Hot air woven was invented by Robert Koch, Louis Pasteur, Robert Petrie, Catton, Jacob Catton. So this hot air woven was invented by Louis Pasteur. So Robert Petrie introduced Petrie plate, Koch postulates, Jacob Catton, father of soil solarization. HEPA filter, high efficiency particulate air. This filter is actually present in laminar airflow chamber, which is used to provide a sterilized air by method of filtration. This HEPA filter size is generally 0.3 micrometer, 0.5, 0.6, 1.0. So generally the size of the HEPA filter is 0.3 micrometer in diameter. So the smallest plant pathogenic agent: virus, viroid, virusoid, and virion. So far the world's smallest plant pathogen is viroid. Generally the viroid is 100 times smaller than that of a virus. Generally 10 times to 100 times smaller than that of virus. This is the smallest plant pathogen that is discovered so far. Nomenclature of a fungi is governed by. So general nomenclature of fungi is governed by ICTV, ICNV, ICNB, ICBN. So ICBN is the governing authority that takes control of this nomenclature of fungi. International Committee on Botanical Nomenclature. ICTV means International Committee on Taxonomy of Virus. So before it was named as International Committee on Nomenclature of Virus 1966. That is name changed to International Committee on Taxonomy of Virus in 1973. So for fungi ICBN, for virus ICTV. So cabbage yellosis is caused by, generally cabbage yellows is caused by generally phytoplasma. No. This cabbage yellosis is caused by fungal disease. Astrellosis, those are all caused by this phytoplasma. But when it comes to cabbage yellosis, it is caused by fungi only. So these are all the answers for reference. For further details and for further preparations and various other kinds of things, students can refer this book, A Vision into Plant Pathology, complete student version. Further doubts and for further classification and guidance, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.